Okay, last part of cellular respiration is oxidative phosphorylation. We're going to divide oxidative phosphorylation into two parts. We're going to divide them into an electron transport chain, like the generic one we talked about. We're going to show it in a very specific way. Um, and then chemiosmosis or chemiosmosis, depending upon who teaches you how to say that word. So remember, electron transport chain, we're going to shuttle those electrons. We're going to transfer them from molecule to molecule so that we can get a little bit of energy off of them. And those molecules are actually going to be proteins. We're going to move them from one protein to another inside the inner mitochondrial membrane. And when we do that, we're going to use them to pump hydrogen from the matrix to the intermembrane space, and we're going to create a gradient. We're going to create a whole bunch of buildup of hydrogen because we're then going to use that hydrogen pump to make ATP. Okay, so here we are, just like this. So I've got this portion right here that is the electron transport chain. This is where we're going to move. We're going to pick up electrons from NADH. We're going to move them through this protein. We're going to transport them. Each one of these purple guys here is a protein. Okay, they're specifically called cytochromes. So these proteins that are embedded into this plasma membrane within the mitochondrion are called cytochromes. And what we're going to do is we're going to move through each of these, and these big integral ones, these guys are actually going to pump hydrogen out, okay, out from here, out against their gradient up here. And what they're doing is we're using the energy from the electrons. So as we transfer them from NADH, we transfer those electrons in across this protein to cytochrome Q. From cytochrome Q, we go to number 3. From number 3, we go to cytochrome C to 4. And our end receptor down here is going to be oxygen. Just like I had told you before, we're going to have oxygen. So what we're doing is we're taking hydrogen ions from a lower concentration and we're using the energy to force them up here to a higher concentration area. And the reason we do that is because of chemiosmosis. And that's the second part of oxidative phosphorylation. And what that does, and I'll show you this pump in detail, but what's going to happen is we're going to use the energy of this proton, of this hydrogen, and we're going to move it through this pump, this specialized protein called ATP synthase. It's really all it is it's just a specialized protein. And the energy that's going to come through here, we're going to use to phosphorylate ADP, and we're going to get ATP out of it. So let's look at that. Okay, so after glycolysis and citric acid cycle, we've got all that NADH and then FADH2, and all of those guys are going to be really where most of the energy was extracted from the food we ate. That's what it mostly gets converted into, is NADH and FADH2. And then those two electron carriers are going to donate electrons to the electron transport chain, and then in the last half of oxidative phosphorylation, we're going to do ATP synthase. Here's your bolded word for cytochromes. So the main components of that electron transport chain, okay, on that inner membrane, those guys are called cytochromes. And they're it's basically a multi-protein complex. When we drew it out, it looked like there was just one. But in that inner membrane and those crystal folds, there's dozens, hundreds of those running all along that membrane. And that's why that membrane is folded, to create that increased surface area to give us more room to have the electron transport chains and the ATP synthase. Okay, the end receiver of those electrons, the same as we said before, that end receiver is going to be oxygen, and then we're going to pick up hydrogen and make water. Okay, so here's just isolating it out of the membrane and really blowing it up. We're going to pull the electrons off of NADH. That'll leave NAD, who's free to go get 2 more. And then we're going to shuffle these electrons through this transport chain. And all along the way, what we're going to do is we're going to move hydrogen ions. Notice we're not producing any ATP here. There is no ATP production that's happening. None. Nada. Zilch, nil, none. The end receptor for those electrons is oxygen, who then hooks up with hydrogen, and we get water. So the thing about this is if there is, If there is no oxygen here present, if we have moved to an anaerobic situation, that's going to be bad for the mitochondria in general. But if that's happened, there's no electron transport chain. All of this stops because these electrons have nowhere to go. And so we can't continue to run this chain. It's like being backed up. So if we can't continue to run it, then we can't harvest the energy from the electrons, we can't pump the hydrogens, and we're going to stop ATP synthesis simply because we don't have oxygen available for the transport chain. Okay, so we're going to move that hydrogen across from the mitochondrial matrix into the intermembrane space, just like we said, and we're moving those hydrogen ions against their gradient, up their gradient. So we're making a higher concentration of hydrogen out in the intermembrane space. And then what's going to happen is, because I have this big buildup of those ions, they want to diffuse. They want to go from higher concentration to lower concentration. And the channel through which they're going to do that is going to be ATP synthase. And that exergonic flow of hydrogens through that pump, through that specialized protein, is going to generate the ATP. So 2 words that we've got to get, okay? Chemiosmosis, and then I think there's one coming up in a couple of slides called proton motive force. Chemiosmosis is using the energy that we created with the hydrogen gradient to do work. So chemiosmosis is a process, okay? It's not a destination, it's a process, and it's the process where we're going to use the energy created in the gradient, right, so that we can do cellular work. It looks like this. So here's my intermembrane space. Here's the mitochondrial matrix. We've been pumping hydrogen out here, pump. Got all this extra hydrogen out here. And this is actually what it really looks like. So the hydrogens filter in here. They end up on this rotating sort of piece of protein, okay? As they rotate around, they end up, there's an internal rod, and they cause this catalytic knob to go. So here's an enzyme protein here. And what ends up happening is, as this spins, we phosphorylate ADP and create ATP. So we're going to create a bunch of ATP here in the mitochondrial matrix. The other thing that happens is this hydrogen gets released off of the upper rotor unchanged. And so it's here in the matrix available to be pumped back out to the intermembrane space. It's kind of cool. Here's that other vocab term I mentioned, the proton motive force. So the proton motive force is the gradient. Okay? The proton motive force is the gradient. It's that potential energy we've created by pumping the hydrogen ions against their gradient. So the proton motive force is that potential energy that we've generated, or that we've created by generating this big concentration gradient. The work that we get, the process of using that energy, so not that we made the energy, not that the energy exists, but using that energy to do work, that's chemiosmosis. Okay. So again, just a visual, just to help you pull all of that back together. We're going to harvest these electrons off of the electron carriers. We're going to use some energy from there and shift around these proteins. We're going to pump hydrogen against its concentration to create a large concentration gradient of hydrogen out here. This large concentration of hydrogen out here is the proton motive force, because it has a lot of potential energy. We're then going to use that potential energy, and we're going to use it to drive the specialized protein ATP synthase. That's how we're going to generate ATP. And the process of using this proton motive force, this potential energy, using it to do this work, is called chemiosmosis. All right, so how are we going to account for all of this? Most of the energy goes in this sequence when we talk about cellular respiration. It goes from glucose to NADH or FADH2 to the electron transport chain, to the proton motive force, to ATP. So from where we start with glucose to where we get ATP, there's a bunch of steps in the middle. About 40% of the energy in a glucose molecule gets transferred into ATP. And we make one glucose molecule makes roughly 38 ATP. That's a pretty good deal. All right, so you need inputs and outputs for oxidative phosphorylation. This one's pretty easy. All I put in is NADH and FADH2. What I get out is the ATP. There's the visual if you want to count it. Okay, so the alternate option to cellular respiration is fermentation. So most cellular respiration requires oxygen, right? We said that. The glycolysis portion doesn't. I mean, oxygen can be present, but it isn't required. So glycolysis can produce the ATP whether oxygen is there or not. So glycolysis can be considered aerobic or anaerobic. I typically think of it as anaerobic because that's my way of remembering that, oh yeah, this can happen even though oxygen isn't there. But in those situations where glycolysis starts, when we get to the end of it, when there is no oxygen, after glycolysis has done its thing, right, and we get that pyruvate, if there's no oxygen present, then we can't move on. We can't go through the mitochondrial membrane. We can't start cellular respiration in the mitochondrion because that requires oxygen for the end of the electron transport chain. So what happens instead is glycolysis hooks up and moves into a different process. It moves into the process of fermentation or anaerobic respiration to produce ATP. So if there's no oxygen present, then we still do glycolysis like normal, but we don't go into citric acid or Krebs. We move into fermentation. So what happens here in anaerobic respiration is we use an electron transport chain with an electron acceptor, something other than oxygen. Okay? So in anaerobic respiration, instead of having oxygen as our end receptor for the electrons, we'll use something like sulfate. What fermentation does is it uses phosphorylation. So instead of using an electron transport chain at all, it doesn't. Fermentation doesn't have an electron transport chain. What it has is the ability to do phosphorylation to generate the ATP. Okay. So fermentation is going to be glycolysis plus some reactions that regenerate the NAD+, and then it can reuse the NAD+ to run glycolysis. So two common forms of fermentation are either alcohol fermentation or lactic acid fermentation. Alcohol fermentation is a two-step process. We take pyruvate and we convert it to ethanol, but it takes 2 steps to do that. The first step, we're going to release carbon dioxide. So this is the process, this alcohol fermentation, this is the process that yeast use in the brewing and the winemaking and the baking. This is my kombucha process that I make. So here's alcohol fermentation. We take glucose, we run it through glycolysis, same as before. We get out our pyruvate. Okay. What ends up happening is that the carbon dioxide portion gets cleaved off of the pyruvate. We get 2-acetylaldehyde. Okay. And then that gets converted into ethanol. Okay, so in this process, we're only making 2 ATP. This is not a big energy ATP deal for us. In lactic acid fermentation, that pyruvate is reduced to NADH. We're going to get something called lactate as an end product, and we don't give off carbon dioxide. So lactic acid fermentation, we do this in some fungi, some bacteria that we use to make cheese and yogurt. This also is what happens in human muscle cells. We predominantly think of skeletal muscle cells, but so this is when we generate ATP when oxygen is scarce. Not that oxygen is scarce in our environment, it's just that it's not there in the cells. We've depleted what oxygen we had there, and we're not respiring fast enough to keep up with the amount of oxygen we need to produce the amount of ATP being used. It's a one-step process, and it looks like this. So we go from glucose through glycolysis, we get pyruvate, and that gets immediately converted into lactate. Okay. How do they compare? How are they the same? So both processes use glycolysis to oxidize glucose or other organic fuels into pyruvate. So they both do glycolysis. The processes have different final electron acceptors. So an organic molecule like pyruvate or acetaldehyde is what we use in fermentation, and oxygen is what we use in cellular respiration, in the aerobic cellular respiration. Cellular respiration is going to produce 38, roughly, give or take, 38 ATP per glucose molecule. Fermentation is only going to produce 2. So really, if you're talking about getting ATP out of something, cellular respiration is the way to go. But if we're talking about 2 ATP or no ATP, then and if you have a lack of oxygen, then certainly fermentation is your better option. Which brings me to different mechanisms employed by different organisms. So we have things called an obligate anaerobe. And obligate anaerobes carry out fermentation or anaerobic respiration. But obviously, because it's an anaerobe, they do it without the presence of oxygen. Now, the fact that these guys are obligate, they are obligated to be without oxygen, which means they don't survive in an environment that has oxygen present. So obligate anaerobes require there to be no oxygen, otherwise they expire. Facultative anaerobes are different. Facultative anaerobes can survive in either situation, whether there's oxygen or not. They have the ability to produce ATP either through fermentation or through cellular respiration. So this is going to be the yeast and a bunch of our bacteria. They're okay either way. Oxygen's there, great, we're going to run cellular respiration. No oxygen present, that's okay, we'll just do fermentation. So they have that option as well. For these guys, for the facultative anaerobes, the big key question is going to be what is pyruvate. So we get to pyruvate, and it's basically the proverbial metabolic fork in the road. At pyruvate, the question is do I have oxygen or I don't? If I don't have oxygen, then I'm going to go to fermentation. If I do, then I'm going to go to cellular respiration. Organisms like you and I, most of the mammals we know, we're obligate aerobes. Okay? No "and" in front of that. Obligate aerobes, which means we require oxygen. We don't survive in a non-oxygen environment. Okay? So we need that oxygen presence in order to generate ATP. That's just the facultative anaerobe juncture at pyruvate. Which catabolic pathway am I going to take? Am I going to go to fermentation or am I going to go to cellular respiration? Okay. So here's a little bit, because I've told you again and again and again, we keep touching on evolution, right? So the significance of glycolysis is pretty big evolutionarily, because it happens in pretty much all the organisms. Just about everybody does some form of glycolysis, and that's really significant. There aren't a lot of processes that all organisms do. What we believe is that glycolysis probably evolved in the ancient prokaryotes. Before there was oxygen in the atmosphere, glycolysis started to exist. And then as organisms developed and as the atmosphere changed, then so did the pathway. Glycolysis and citric acid cycle are both major intersections. There's a lot going on there, and they lead to various different catabolic and anabolic pathways. So it's not like this is an end stop for these organisms. They're actually bridging points. Glycolysis, even though we've been talking about it working with glucose, it'll take a wide range of carbohydrates. It doesn't have to be just glucose. We can also use proteins if we digest them into amino acid groups, because those amino groups can feed glycolysis or the citric acid cycle directly. So we can start with proteins if we can break them down. Fats that are digested into glycerol, okay, we use that glycerol in glycolysis, and the fatty acids are used in generating acetyl-CoA. So even fats are important to cellular respiration. So how we break them into acetyl-CoA is we do something called beta oxidation. I don't care that you remember beta oxidation at all. In fact, if you don't, I wouldn't even bat an eye. I would know that I get my acetyl-CoA from fatty acids. Okay. When we oxidize a gram of fat, we're going to get twice as much ATP as that gram of carbohydrate that we started with. And that looks something like this. So we can start with any of these, and they're going to feed into the system at different points. So if I look at this purple line, I can start with proteins. We can break those proteins into amino acids. They can fit in at the pyruvate, at the acetyl-CoA stage, or the citric acid cycle, and we can still finish out. Carbohydrates, obviously, we've talked about. We've got the sugars. We can undergo glycolysis. Or if we don't have a simple sugar like that, we can start here and form into glyceraldehyde 3-phosphate, or the fats. The glycerol can come in right here. The fatty acids come in down at acetyl-CoA, and we're still going to run this pathway. Okay. All right, feedback inhibition. So we talked about negative feedback loops early on. So that's how we're going to control cellular respiration. That's how we're going to run this mechanism. So if we're in, and the thing about, if you'll remember, ATP is not very stable. It doesn't just store and build up well in cells. You can't just stockpile your ATP and have it build up and then it's good and ready to go, so that when you go to run your marathon or your whatever it is you're doing, you've got all this extra ATP hanging out, because it just doesn't work like that. ATP is not one of those molecules that likes to just hang out and be stable and wait. So as that concentration of ATP begins to drop, cellular respiration speeds up. When there is plenty of ATP, and that doesn't necessarily mean a lot of it, but when there's plenty of ATP around, then cellular respiration is going to slow down. We don't ever create a giant stockpile. Okay? And how that works is this. So when I have lots of ATP, I'm going to get an inhibitory response on the phosphofructokinase, the enzyme right here up in the glycolysis pathway. Okay. And then the other thing that can happen is when I have plenty of citrate off of the citric acid cycle, it'll have the same effect. It'll halt the phosphofructokinase. AMP, which is one from ADP, so ATP is adenosine triphosphate, ADP is adenosine diphosphate, and then AMP is adenosine monophosphate. When I have a bunch of monophosphate around, when I have a bunch of AMP, that means I've cleaved off one and made ATP, I've cleaved off another and I've made from ADP to AMP, and that's a trigger of, hey, I don't have enough ATP around. And so what ends up happening is that AMP presence triggers or stimulates phosphofructokinase to drive out more ATP. All right, that's the end of our slideshow. So I'm going to call that here. We'll publish it. I'll get it out for you guys. That's Chapter 9, Cellular Respiration. If you've got any questions, shoot me an email. Thanks.